Good afternoon, and welcome to the CRISM Therapeutics Corporation Investor Presentation. Throughout this recorded presentation, investors will be in listen-only mode. Questions are encouraged and can be submitted at any time via the Q&A tab situated on the right-hand corner of your screen. Simply type in your questions and press send. The company may not be in a position to answer every question it receives during the meeting itself. However, the company can review all questions submitted today and publish responses where it's appropriate to do so. Before we begin, I'd like to submit the following poll. I'd now like to hand you over to Andrew Webb, CEO. Good afternoon, sir.
Good afternoon. Welcome to today's CRISM Therapeutics Corporation. Thank you very much for taking the time today. My name is Andrew Webb, and the purpose of today's presentation is to update you on the significant progress we've made over recent months. Those of you who may recall the last update was in September. And clearly for today, it's in the context of seeking additional funding from our existing shareholders through a retail offer which was published. This is a follow-on from a successful and oversubscribed placing which the company did earlier in the week. So to my way of introduction, just a refresh, CRISM Therapeutics, we are a drug delivery company working on how we can improve, importantly, the safety and efficacy of chemotherapy for cancer patients. If I may, just by virtue of an overview, just as a refresh, we are ourselves as a platform drug delivery company, so we believe the technology that we have, which is named ChemOseed, will be available to and accessible to a number of solid tumour indications. What we're doing at the moment, as always, to give you a run into the clinical trial plans that we have. But just quickly on the technology itself, it's termed ChemOseed. ChemOseed was the innovation of our chief scientist, Professor Chris McConville. He'd been working in the oncology space for some time and realised there was a way we could much improve the treatment of patients. And this is really the clinical performance of chemotherapy is really very much what we're about. And I'm pleased to confirm the novelty of the technology, and this was confirmed through the grant of our European patent last year. So our lead product is ChemOseed, and it's combined with a chemotherapy drug called irinotecan. Irinotecan is a well-known, well-understood drug originally out of the Pfizer portfolio, and is currently still used in first-line treatments in other indications, including bowel cancer and in second line for brain tumour. And brain tumour is the indication we're starting our journey with. Brain tumour, many people aren't aware, just as the seriousness of it, it's the largest cancer killer of children and young adults under the age of 40. Sadly, the standard of care remains unchanged since 2005, and for those on the journey we're starting in the recurrent phase, there is no standard of care. So it's most important that we progress as quickly as we can with this indication. And what was really pleasing is that the clinical trial I'll come on to describe has been approved by the MHRA, and we are scheduled to be dosing our first patients in the first quarter of next year. So by way of a quick refresh on the technology, this is termed ChemOseed, and on the tray on the right you'll see the small yellowish coloured cylinders. Those are the ChemOseeds, and they're racked out in a plate, and this is how they'll be presented to the neurosurgeon for implantation. And the cartoon just to the lower left shows the implantation of the seeds, and they're sunk into what we refer to as the resection margin. So once the tumour has been removed, there's sadly always going to be a nominal amount of residual tumour, which sadly is inoperable. It may be sort of large pieces, it may be through microscopic amounts of tumour, but sadly it's this section from which a patient will progress and the tumour does return, which is the real challenge. So on the right you'll see a schematic of how the implantation is done. The pen-shaped instrument you can see there, termed the ventricular catheter, it's a standard piece of equipment that uses during neurosurgery, and it's used to move the tissue around for different purposes, and in our case it will create that space that we can sink the implant in there, and what we're doing is effectively directly treating the margin. The kind of like a roadmap to the right, that indicates the spacing, so we need to take account of the diffusion distance of the drug within the tumour margin, And this is done by spacing the seeds within that 6 millimeter distance between each. And that ties in with the size of the implant. So they lay down the bigger space, drop the implant in. It's very simple, straightforward. The neurosurgeon has a limited amount of time on which to conduct this procedure, and we are estimating on their guidance somewhere around 30 seconds each. Look on the left on the regulatory side, it's very pleasing. We are being seen as a reformulation, so we don't have any medical device regulations to deal with. So this has been a very clean application with support from the MHRA. One piece of news we received recently, which was very pleasing, is that a very reputable group at the University of Edinburgh ran a machine learning AI program to identify suitable drugs for the treatment of glioblastoma. The system, which has now been peer-reviewed and published, screened some 12,000 drugs, chemotherapy drugs. That was produced a short list of 3,500, and they finalised a selection of 3 drugs they felt showed the most potential for GBM. And irinotecan, the active metabolite SM38, is one of those 3 drugs they identified. So I believe they are excited to see the progress that we make and when we can have a clinical validation of their work. Success in this will open up other indications, and those are listed on the left-hand side. So what's happened since we last spoke? Well, the key thing is that the MHRA has approved us for a phase 2 registration trial, and that was approved back in September. Want to note this is an open label study, so very importantly that we can have an open dialogue with the regulator, and we'll be able to keep not just patients, but also shareholders and others up to date on progress when we first dose our patients, and how the patients are tolerating the treatment, and certainly going to report an efficacy signal. The last funding round was key. We were able to initiate the manufacturing of the clinical batch of chemosine, and that is underway. And importantly, we appointed the clinical research organisation to set up and manage the trial. And that work was initiated back in August, and that's underway, and we look forward to reporting as the sites begin to open. So this is really a pivotal moment for the company, and our first patients will be dosed early next year. And to say the open label design, an open label, just to be clear, this means that we can, both the patient and the physician know that the patient is getting treated and what treatment they're getting. And what we will look to see, and this is the reason why we've done a small extension on the part 1 of the trial, that once we've done the dose escalation, which I come on to describe, we will be able to report an efficacy signal. This clearly gives us potential for a conditional market authorization. And to be clear on this, what it means for us is that we will have a commercial product at that point. I think that puts us in a most unique situation not just in the brain tumour space. So why it matters? We're on a rapid regulatory pathway with the MHRA. The program is called ILAP. You may remember we secured what's called an Innovation Passport, which is the MHRA's endorsement for the rare disease treatment. ILAP is their equivalent of orphan drug post-Brexit. It's the Innovative Licensing and Access Pathway. And this brings the potential. This is the framework we're in for an accelerated approval. And we've had very strong and supportive engagement on conversations with the MHRA. Why does it matter? Well, I think I've already touched on this, and many of you have heard me before will understand the recurrent glioblastoma, there is no standard of care. But the support is out there that we should be able to leverage technology to make good progress here. And it's this ILAP program that's so important. What is really exciting is that we are, in that framework, we get access to what's termed Project Orbis. This is an FDA program. And it's to take up, and it's specifically focused on oncology and also on rare disease. And brain tumour comes under that category. So what it means is that the FDA, there will be early insight for review of the data, and this ties in other jurisdictions such as Brazil, Canada, Switzerland, Australia, and others to also participate. So what's happening today? Why is this so important? And in cancer treatment today, many drugs have limited distribution. So what happens is cancer therapy is given. It tends to be given either as an injection, infusion, or a tablet. And often in most cases, less than 1% actually reaches the tumour itself. And some of it actually has very little absorption into the tumour. So we believe by implanting the drug contained within an implant directly into the tumour tissue, we get the best chance of a safe and efficacious dose. And with this, it's important that side effects, given the toxicity of these drugs, has a major impact on quality of life, and that is something that we hope to address. In an earlier study, the circulating levels of drug were some 1,000-fold less than you'd expect on an infusion. So we have the reasons to be optimistic here. Other treatments have come and gone, and largely it's due to the side effects. And the side effects do clearly then require treatment, and that's additional cost to the health service. And that's something that is part of our agenda. So clearly, I'm sure it's well understood why this matters clearly clinically. The implant is directed, and it's this resection margin I touched on, which is where glioblastoma, where the tumour will recur. And then the challenges that other drugs have is that what's called the blood-brain barrier. The brain has a protective mechanism which makes it very, very difficult for drugs to cross into the brain. And that's the reason why treatment options are severely limited at this point. We gave the example also from the surgeon's perspective, from the images earlier. It does make it very easy for the surgeon. The implantation time is important. We were being told that whatever procedure we introduced must take less than 40 minutes. The NHS under its constraints, the surgeon needs to ideally perform 2 surgeries a day. If we run on for too long, that limits the number of procedures they can tackle, and clearly that's not acceptable for it to be reduced. So another reason I think, just the final point, is why we believe this treatment will be important. It's one of the rare times when we actually can start treating a patient immediately post-surgery. Up until now, in the standard of care, a patient may wait some 4, even 6 weeks, maybe longer, for recovery from the surgery prior to any treatment, which is typically chemoradiation, as chemotherapy with the drug temozolomide, and radiotherapy. And over those periods of several weeks, that residual tumour can grow back really very aggressively. So we believe when the tumour bed is at its lowest volume, we stand the best chance of success. Just to quickly reflect, some of you may have seen this before. It's on our preclinical results. This was done using what's called a patient-derived xenograft. So this is a human glioblastoma cell line. It's very aggressive. It's implanted into a mouse, and we were then using the chemoseed treatments on them. So if you look at the graph on the left, the black line is the placebo with no treatment at all. As you can see, the condition post-surgery is very, very serious, and the mice have a very short survival. The centre 2 lines, the first one is intravenous dose of the drug immunogen we're using, and that the improvement there is typical, as is temozolomide, the blue lines to the right. Clearly, it does show an improvement, but it is more managing the recurrence than treating it. And here you can see the survival line in red, where the mice were treated with the chemoseed treatment containing irinotecan. So this was some really, really positive news. This is 148 days, which is the ethical limit. So at that point, it's no longer permitted to continue, and the mice, particularly on the chemoseed treatment, were euthanized at that point. And this is where we got really enthused about the data. The right-hand pair of panels, you'll see this is treated with the standard care temozolomide, and you can see from when the first mouse passed at day 41, you can already see the tumour is recurring. It does put some delay, but all of those mice died through the tumour, and the red image to the right of the images of the brains does show that effect. If you look to the paired panel on the left with chemoseed, there is no recurrence at any stage. The mouse that We lost early on in the study. The laboratory's technician reported that they felt it never fully recovered from the surgery, which makes it quite tricky in this indication. The other 4 showed no disease progression whatsoever. So that really gives us reasons to be confident. I'll go through this very quickly. This is just that we have been done some phase one study. This is some years back. What was the key message from this, without going all line by line, is that the drug was safe. And to inject a chemotherapy drug directly into brain tissue, which is what happened here, they injected over 30 sites in this resection margin once the tumour had been removed. There was no problem associated with it. Typically, in other instances, a drug called carmustine has been used with a vehicle called Glidel. That causes inflammation, swelling, and it impacts wound healing from the surgery. And the patient then becomes susceptible to infection, which is most serious in this situation. So this really gives us reasons to be optimistic about what we have here. I'd say early next year we'll be looking to make that clear assessment. Just as one final point, this phase 1 study was about irinotecan, not about chemosine. This was on a formulation which was injected, which was the predecessor to the chemosine technology that we currently utilise. So this is where it's the important part now. So we're now heading into trial next year, and the funding round that we've been in now enables this and gets us through the dose escalation. We have an opening dose in part 1. We're starting with, as mentioned, recurrent patients. And these are patients who sadly have followed their initial diagnosis. They've had surgery, the tumour has suddenly returned, and they're back for a further surgery. We'll start with an opening dose. This is going to be measured in seeds, so 10 seeds for the first patient. And that is a dose that's within the safe window that we demonstrated in the phase 1 study. We will then progress very quickly through up to 30 seeds, which will be at patient 4, which we believe will be the maximum treatment dose. The dose isn't so much dictated by the amount of drug and implants we're given, but the size of the resection cavity. So clearly, before when that assessment was made through a patient's brain scan, when we remove a larger tumour, we will need more seeds to treat the resection margin. And that's the reason for the variation. But we need to demonstrate that safety at this stage. We will be looking for an efficacy signal, and this is where we've been made aware that there is a very clear appetite for an early approval here, given the unmet need. For these patients, sadly, as I mentioned earlier, there is no standard of care. It's very much a decision between the oncologist and the neurosurgeon as to what course of action can and will be taken. Success in terms of its safety is the key output of part 1. That will lead us into part 2, and then we will hope to then start treating patients who are newly diagnosed. And any efficacy signal in part 2, which is much more part 1, should be much more effective within part 2. And this is market authorization that we're going for, and this will ideally bring us through to a commercial product. The milestones, I appreciate it's not a long list, but actually we're very clear on what we're doing now. And this is where we've been very pleased with the response we've had and engagement from our investors. People that put money in the summer, a lot of them have followed. And a number of new people now are optimistic now the progress we can make as we dose our first patients early into next year. And this is an open label study which gives us a clear and ongoing dialogue with the UK regulator. To say the conversations have been very supportive, and it is that once we show that the treatment is safe, well tolerated, we're then looking for an efficacy signal of the approval. There is a note here. This obviously will be subject to not just recruitment, but also ongoing funding requirements. I know it's a sensitive one, but at the moment that is the money that gives us to invest and to enable this to happen. The trial is mapped out that we only pay for the patients that we test and the clinical trial centres that we use. Every effort, please be confident with the board, is we're making to manage that cash flow as best we can. We are actively seeking non-dilutive funding in terms of grants and other routes, and I'll clearly make notifications on success as those discussions progress. The market opportunity, we're excited about it. We've mentioned before, we estimate some circa 1.7 billion in totality. That's across the brain CNS space. For us, this is to achieve standard care, certainly should be achievable. It is a rare disease, but clearly we need to give patients genuine hope that we can make a difference here. And once we show progress in the brain tumor indication, we do believe, as indicated by the table here, we can start and take this out. And as many of you will be aware, we commenced our second program in prostate cancer, which started back in May. We've had some good early progress on our formulation work with that. The market size and the costings, we're still in work in progress. On here, it just notes in the subtext that we have a nominal estimated of £13,500 per patient treatment. That was based on a nice value of quality adjusted life here, £20,000. The metric usually now is in the range up to 30,000. For rare disease, it could be as high as 50. So the company will make a reassessment of the opportunity here. But we are committed that we make this treatment available for all comers. A number of new treatments which are of great interest coming forward. Some of them may end up being prohibitively expensive, and CRISM is not in the business of fueling this health inequality that we're currently seeing, given the funding challenges that we have with our health system. So our final slide. So here we are. We are ready. We've been talking about this on the journey along the way, and we are in discussions. As you're aware, that the technology that we use came out of Professor Chris McConville's work at the University of Birmingham, and so we remain strong links, and the neurosurgical team there has been a huge support and a mentor for Professor McConville and for the company in how we bring this through. And they worked, our medical advisor was from Birmingham, Professor Garth Cruikshank. Many of you would have heard on my last update in September, and he has very carefully helped craft the structure of the trial, which we just described. So it's dose escalation safely and looking for secondary endpoints in efficacy in part 1, and a quick move into part 2, where we want to demonstrate efficacy for the newly diagnosed. And this is going to be the journey that will start. And in a matter of months, we have to give a short time between each patient to once we understand that each dose level is safe and effective, we decide safe and well tolerated, we can then move to the next dose. And there will be frequent imaging taken on a 4-weekly basis in order that we can very quickly assess the impact. Given, as I mentioned, that the overall survival for patients, and sadly with this disease, is 14.6 months. So we would expect to see and have an understanding of success at an early stage. And this is open label design that permits not just the interim analysis, but it allows that to be reported. And the MHRA have directed us to keep them aware of progress. And it's some approval here. We believe it will very rapidly open up opportunities for other indications. Prostate will be have some further news in the future as that program is at its early stages, but we look forward to keeping you informed along the way. And clearly the plan is that success of the brain tumor indication really should open up other opportunities. So with that, I'd like to end the formal presentation. Your time is valuable, and thank you for taking the time. I'd like to open for any questions that you may have, as directed by Investor Meet Company at the opening. Thank you very much for your time and for your attention.
Andrew, thank you very much for your presentation. If I may just bring back up your camera. Ladies and gentlemen, please do continue to submit your questions just by using the Q&A tab situated on the right-hand corner of your screen. Just while the company take a few moments to review those questions submitted today, I'd like to remind you that a recording of this presentation, along with a copy of the slides and the published Q&A, can be accessed via your investor dashboard. Andrew, as you can see, we have received a number of questions throughout today's presentation. If I please ask you to read out the questions and give responses where appropriate to do so, and I'll pick up from you at the end.
Thanks, Luke. So a couple of questions that came in early on, and I do understand that people are expressing their concern. So they've been disappointed with the recent placing in terms of the effect it has on the share price. The director you see from the last round, we actually have a stake in this. We're aligned with you. The markets are very difficult. The capital markets are very demanding at the moment. There is a reality, and it is the nature of we're in, that the next expectation is that they receive the shares at a discount. I'm pleased that on this round we've minimised the discount compared to the summer round, but I have to say I do share your pain. If it could be different, then we would certainly endeavour to do so. But we do have certainty now on testing patients. This runway, this £1 million, is so important. Clinical trial costs are expensive. We are planned, it's happening, we will be treating patients. And I do hope that the market responds favourably at that point. Further question, when is the first patient likely to be dosed, in January or March? Our timelines, I would say it's more likely to be March at this point. There is no regulatory hurdle in terms of progressing this study. It is now all operational. As you're aware, we don't carry these costs directly internally. We manage these work packages through third parties. Our manufacturing was actioned back in August. The manufacturing partner that we have has twice manufactured batches of the product on our technology transfer when we validated the manufacturing protocol from Professor McConville's laboratory in Birmingham to what's called a CDMO, a contract development manufacturing organisation. These are the people that manufactured the pharmaceutical grade materials. That ran seamlessly. They since made a second batch, which is under GLP, called Good Laboratory Practice for animal studies. That was successful. So we are confident now on the GMP, which is a good manufacturing practice batch, which is the clinical batch. We would receive that materials on time. And they're due into the first quarter of next year, likely February at this point. The clinical trials take time to set up, so that's another reason. So that's in hand at the moment, and we're filing the paperwork at this point. It does take time. It is frustrating, but the databases and the secure service to collect the patient data and record it and report it does take time to set up. So we are looking, I would say, into the second half of the first quarter. But please be assured, this is an exciting part of the journey, and we'll look to keep you informed. Next question, what are your long-term plans for commercialisation, independent launch, co-promotion or full licensing? To be honest, the options are under review at the moment. The UK market is manageable in the first instance. We have opened up discussions with a distribution partner who have a launch program for new drugs. Given the severity of brain tumour and essentially the treatments will take place in what we effectively refer to as centres of excellence. Centres of excellence for glioma, which is the brain tumour. There's probably around 14, 15 directly. Those often have been endorsed by the Tesogel Brain Cancer Mission for their assessment of their ability. They need to be treating somewhere around, I think it's about 75% of their patients need to be high-grade glioma, and these will be our target commercial opportunity in the first instance. A number of these sites will be joining us as part of the trial, so we will have existing relationships on which to continue ongoing supply. It is a small and close network community of neurosurgeons, so we hope the news will travel fast. We will be looking to partner. Quite what form that will take at this point remains to be seen. And now the clinical trials is set up, it's one of my responsibilities now is to map out some more clarity on the commercial model at this point. And again, it's a good question, and thanks for asking. That is the final question I have at the moment. I don't know. I think perhaps I'll hand back to the Investor Meet Company. Thank you.
Andrew, thank you for answering all those questions you can from investors. And of course, the company can review all questions submitted today and will publish those responses on the Investor Meet Company platform. Just before redirecting investors to provide you with their feedback, which is particularly important to the company, can I please just ask you for a few closing comments?
I just want to say thanks to everybody. Your support has been invaluable. The investment is key, and whatever happens, we can take improves the cash runway that we have. The trial is there. It will continue to run at a pace. Once we reach the treatment dose I've mentioned earlier, 30 seeds, we will be treating patients to generate that registration data as fast as we can recruit. And steps are being taken to support the recruitment process aligned with our contract research organisation. So we look to hopefully have early news for you. It will certainly be done with best efforts going forward. But thank you again for your support and engagement and taking the time to listen to the presentation today. I'm very grateful. Thank you.
Andrew, thank you for updating investors today. Can I please ask investors not to close the session, as you'll now be automatically redirected to provide your feedback in order that the management team can better understand your views and expectations. This will only take a few moments to complete, and I'm sure will be greatly valued by the company. On behalf of the management team of CRISM Therapeutics Corporation, we'd like to thank you for attending today's presentation, and good afternoon.